First of all, where am I inside the chloroplast? This is the location where am I, right? I am at the thylakoid here. That's the membrane. So that's the membrane of the thylakoid. This is the inside, and this is the outside. Okay, so again, think of the thylakoid as like something like this, right? This is in, this is out, and obviously outside is going to be what? Outside is the stroma, okay? Now, the membrane of the thylakoid is special because look what it has. First, it has chlorophyll. There is chlorophyll molecules, plus it has special proteins inside the membrane. Those proteins, we usually call them the protein carriers, or usually we call them the just call them for the time, but let's just call them protein carrier molecules, okay? Now, what is the very first event that takes place right here? First event is that the light hits that chlorophyll molecule. See that chlorophyll here? It hits that chlorophyll molecule. As this happens, an electron is excited. What does it mean when I say an electron gets excited here? It means the light gives energy. That energy is good enough to pull an electron out from an atom inside that chlorophyll, out, right? And then that chlorophyll is matched by one of those proteins inside the membrane. Again, what happened just now? Light hit the chlorophyll. One electron was pulled out because of all the light energy that came in. That electron went to a protein carrier here. It's going from one to another. But do you notice what's happening here? As it's moving from higher level, was at very high level in the beginning, and it's falling down and down and down here. Now, usually energy cannot be lost or gained, right? That's like the basic principle in energy. What we gain here from the light is not going to be wasted. What is it going to be used for? That energy that we gain from the light, we're going to use it for something else. What are we going to use it for? We're going to use that energy to pump protons, right? We're going to pump them inside the thylakoid space here inside, right? Again, what have we done so far? We took light energy, we excited an electron, so we just made an electron go into a higher state. Then that electron was run through a circuit or a chain of proteins. And then while this electron is running down, we use that energy for something very useful. We took all of those hydrogen ions from water and we accumulated them, we stacked them inside the thylakoid space, okay? What for? Now, you see, we built up a high concentration of protons inside the thylakoid space. We're going to now let them all go out from here. But while they're coming out, we're going to use them for something even more useful. So again, we built up a high concentration gradient of those protons. While they're coming out, we're going to let them go out from a very narrow channel here, right? This is like you're doing something, if you've ever seen a dam, and we accumulate the water here, okay? While the water is falling down, I can use that for what? I can use that to generate electricity, right? Think of it like a turbine. The protons run through this tiny, narrow channel. While they're running, they're actually doing work. What's the work is you doing here? They are actually used to make ATP. All right? So one more time, let me show you what we've done so far. We took those protons, we accumulated those protons inside the thylakoid. We built a gradient, and then we let them go out through a very narrow channel. This channel, check its name. The channel's name is known as ATP synthase. The name suggests the use of this enzyme. What's this enzyme for? This enzyme is to make ATP. Cell peptides make ATP synthase, an enzyme that makes ATP, right? So I'm making ATP here. Remember the word we took at the beginning of the class? Photophosphorylation, all right? And now that's the thing. We made ATP out from this process, all right? So we're going to give a title to this story, the story that we have right here. What's the title or what's the name of what we've seen here so far? We usually call this chemiosmosis. What does chemiosmosis? Definition. Chemiosmosis is to move hydrogen ions from one membrane to another. You built a gradient gradient of hydrogen ions to make ATP, right? So that's like the official definition of what we have done right here. Again, I'm going to repeat this several times. Just bear with me. It's not that easy at the beginning, but let's just do it one more time. Again, what we are doing here, we are in the thylakoid, okay? We have in and out. What do we do first? First thing we do is that you need light, okay? The light is needed for what? The light is needed for photolysis. Water gives me something valuable. What is the thing that water gives me? Water gives me like hydrogen ions and electrons. The electrons go into a higher state here, okay, like this. Then they're carried by a bunch of proteins, all right? And then these electrons will eventually get where? Those electrons will eventually get to the NADP here, see? So I need like a final acceptor who's going to take all of these electrons at the end, and NADP takes them, right? Same thing here. Those electrons, while they're like going through this circuit here, what are they going to do? They're going to help me to accumulate hydrogen ions inside that thylakoid, right? So that's the first thing. I call that the electron transport chain, okay? And then all these hydrogens, what are they going to do? They're going to move through this channel I have here, and we're going to use that for the making of ATP, okay? Again, what's the role of the NADP here? The role of NADP is to take two things. It's going to take that electron, final acceptor. It's going to take that hydrogen ions from here. You know what happens if I have electron and hydrogen ions? Those are hydrogen atoms. And that gives me what? That becomes what? That becomes the actual NADPH, right? Let me show you again. Put that here. Okay, and it's becoming NADPH. Okay, we have another question. So the electron attract the hydrogen in. Very good. The electrons are on the membrane. The electrons are not attracting it directly. No, the electrons are giving me energy. This energy helps me to pump the hydrogens in. All right. It's not about negative, positive attraction. No, it's about the electron. We're at a higher state, and while they're moving down, it's providing me with enough energy to move those protons or hydrogen ions inside the thylakoid. So let me show you this one more time. What do I have here? So I have this process. First thing we do, we absorb the light. Okay, so we're absorbing the light energy by that chlorophyll. Then this causes electrons to jump to a higher state. The electrons run through a chain. This chain is needed for what? This chain is needed to pump protons inside the thylakoid. All right? Now, as you can see here, I have like two chlorophylls. I have like one chlorophyll molecule here on the left. I have another chlorophyll molecules on the right. Why do I have to have two? Actually, you need just to, every now and then, you need to push those electrons to give them like a little push, to give them like a little bit of energy. So I have what I call here photosystem, call it photosystem 2. What's that photosystem 2? It's like a chlorophyll molecules found at this side of the thylakoid membrane. And I have what I call photosystem 1. Again, why do I need 2? Because I need to give more and more energy to those electrons. Again, the end result is what? The end result, look what I've done here. I have like accumulated a lot of hydrogen ions. What will all of these hydrogen ions now do? They will run through that enzyme, ATP synthase, and they're going to be used to make ATP, okay? This type of, like this process or this way of doing photosynthesis, we call it, the one I've just showed you here, we usually call it like cyclic photosynthesis. Why is it called cyclic? It's known as cyclic because, as you can see, the electrons, so here, are like running in a circle. Let me show you here. So the electron basically went from, remember what we call this? Photosystem 2. The electron went like this through those proteins. Then we have photosystem 1, all right? Where will it eventually go to? It gets to the NADP. All of these protons have been accumulating here. What am I going to do to all of them? we will build them up, right? And eventually, we will let them go out through tiny channel known as ATP synthase. When they go out, they will help me to make that ATP, right?